You know, when the Pfizer-BioNTech COVID-19 vaccine got authorized back in December 2020, it really did feel like a scientific miracle, didn't it?
Absolutely.
I mean, going from just a genetic sequence to sticking needles in arms in less than a year, that was just unheard of.
Completely unprecedented speed.
But, here on the deep dive, we know miracles aren't usually just poof out of nowhere. They're built meticulously on decades of science.
That's exactly right.
So our mission today is to kind of pull back the curtain, really deconstruct how this amazing achievement actually happened.
Yeah, and for this deep dive, we're digging into a really detailed technical breakdown of the vaccine's biomedical architecture. That's the term our source uses.
Biomedical architecture, I like that.
We're going to explore how all these different mature fields of research, things like basic coronavirology, really advanced mRNA biochemistry, and super sophisticated nanomedicine delivery systems, how they all kind of intersected at just the right time.
And the pandemic was the spark, the catalyst.
It was the catalyst, absolutely. It forced this incredible integration and, super rapid application.
So for you, the listener, picture this like looking at the blueprints for some incredibly complex, like high-tech machine. We're going to peel back the layers, understand each critical piece, and the really clever engineering choices behind them. By the end, you'll have a really solid grasp of the specific techniques, the science that made it all possible.
You really will.
Okay, let's start right at the beginning then. The whole global race to make a vaccine for SARS-CoV-2, it all hinged on one absolute must-have, getting the virus's genetic code. Sounds simple, maybe, but it was the critical first step.
It really was, and it literally started right there in the clinic. With biological samples, those are nasopharyngeal swabs or pharyngeal swabs collected from people showing symptoms.
Right.
Take the very first confirmed U.S. patient, for example. Their samples were collected maybe 3 days after symptoms started. And the initial confirmation, saying, yes, this is SARS-CoV-2, that was done using RT-PCR, real-time reverse transcription polymerase chain reaction.
RT-PCR, yeah, we heard a lot about that.
We did. It basically detects specific bits of the virus's RNA. Think of it like a super-sensitive search engine for molecules. It takes the viral RNA, converts it to a more stable DNA copy, makes millions and millions of copies of that target sequence, and then a fluorescent probe lights up if it's there.
And there's that C-value thing, too.
Yeah, because the cycle threshold or CETA value. A lower CETA, like the 18 to 20 they saw for that first U.S. patient, that tells you there's a lot more virus in the sample, a higher viral load.
Oh, okay.
And having that high viral load was really crucial for the next step, trying to actually isolate the live virus.
Right, because the RT-PCR just tells you the genetic material is there, like finding, footprints in the sand. It doesn't give you the actual creature.
Precisely.
So to study it, to make a vaccine, you needed the whole live replicating virus. How'd they get enough of that?
That requires a process called in vitro propagation. Just means growing it in glass, in a lab dish, not in an animal.
Okay. High security labs, I assume?
Oh, yeah, absolutely. High-level biosafety conditions, PC3 or BSL-3 labs. The standard method is to inoculate a monolayer of cultured cells, basically a single layer of cells growing flat in a dish.
And what kind of cells?
The workhorse for this, for decades really, has been the Vero E6 cell line. They come from African green monkey kidney cells.
Monkey kidney cells? Why those?
Well, they're widely used in virology because they're super susceptible to a lot of different viruses. But here's the really key part. Vero E6 cells lack a functional interferon system.
Interferon. That's like the cell's burglar alarm, right? It's natural defense.
Exactly. Interferons are the first responders. They warn nearby cells. They try to shut down viral replication. But Vero E6 cells, they can't sound that alarm.
So the virus just walks right in.
It basically rolls out the red carpet. Without that interferon system, the cells can't fight back effectively, which lets SARS-CoV-2 replicate to really, really high levels, or titers. And that's exactly what researchers needed to generate a good viral stock for study.
How did they know it was working?
They'd watched for something called cytopathic effect, CPE. Visible changes under the microscope, the cells start rounding up, detaching from the dish, eventually breaking apart, usually within maybe 2 or 3 days.
Oh, cool.
That's the first visual clue. Then they'd use RT-PCR again to track the viral load going up, the CETA value going down. Once they saw widespread CPE and confirmed a high viral load, they'd harvest everything. That harvest is your viral isolate or viral stock.
So the fact that labs all over the world, Australia, Canada, US, Italy, isolated this virus almost at the same time, that wasn't some sudden breakthrough. It was more like everyone had the same toolkit ready to go.
You absolutely nailed it. That Rapid global success wasn't about inventing new isolation methods on the spot. It was a direct payoff from this globally established, decades-old infrastructure for virology research.
Wow.
The widespread availability, the standardized use of Vero E6 cells, the common culturing protocols, it essentially created this sort of de facto universal platform.
So everyone was speaking the same language methodologically.
Precisely. And that was a huge accelerator. It meant individual labs didn't have to waste precious time reinventing the wheel, developing and validating their own techniques. It allowed for fast, consistent, reproducible generation of high-quality virus stocks. Absolutely indispensable.
Okay, so now they have lots of live virus, the final piece of this foundation, getting the full genetic sequence.
Yeah.
How do you go from virus in a dish to a digital file, that blueprint?
Right. First step is extracting the viral RNA from that isolate, usually using standard commercial kits like from Keegan. Pretty routine stuff.
Okay.
Then that purified RNA goes into whole genome sequencing using these powerful next-generation sequencing or NGS technologies.
NGS.
Right.
The basic idea is convert the fragile RNA into more stable DNA copies, chop that DNA up into lots of small fragments, and then the NGS machine reads millions of these short fragments. These reads all at the same time.
Like reading millions of tiny snippets of the book simultaneously.
Exactly. And then powerful bioinformatics software acts like a super smart librarian, taking all those overlapping snippets and assembling them back together like a giant puzzle into the full continuous genome sequence.
What the big moment?
The pivotal moment was January 10th, 2020. Chinese researchers published the first complete SARS-CoV-2 genome Wuhan Hu1, just under 30,000 RNA bases long, onto public databases like GenBank.
January 10th, that feels so early now. So just putting that sequence online, you said earlier that fundamentally changed how vaccine development happened. Explain that digital leap again. What was the big shift? It was a total paradigm shift. Think about the old way. A new virus emerges. You'd have to wait weeks, maybe months, for labs to get actual physical samples of the live virus, carefully grow it up under high containment, purify it. Then you could start the slow, laborious process of designing and testing vaccine candidates.
Slow, dangerous, complicated.
Right. But with the digital sequence available online, researchers could download it on January 10th and immediately start designing. They could synthetically create the DNA template needed for an mRNA vaccine without ever needing to handle the live virus themselves.
Oh.
It completely decoupled the initial design phase from needing a physical virus sample. It's like shifting from needing to ship actual car engines around the world to just emailing the CAD files.
That makes sense.
That digital to synthetic workflow was arguably the single biggest reason for the incredible speed we saw in the subsequent vaccine development.
Okay, sequence in hand digitally at least. The next huge decision. What part of the virus do you target? What do you teach the immune system to recognize? And this wasn't just throwing darts at the viral genome, right? This was rational design.
Oh, absolutely rational. And space. So the SARS-CoV-2 virus particle has 4 main structural proteins: spike S, envelope E, membrane M, and nucleocapsid N.
SEMN. Okay.
From day one, there was almost universal agreement among researchers.
Yeah.
Target the spike protein, the S protein.
Why the spike?
This decision wasn't a guess. It was built directly on more than a decade of intensive research into 2 other deadly human coronaviruses: the original SARS virus from 2003 and MERS, Middle East Respiratory Syndrome.
Right, the predecessors.
Exactly. And that prior work had clearly shown the S protein's absolutely central role. It's this big glycoprotein sticking out from the virus surface that creates that corona, the crown-like appearance.
Ah, hence the name.
Precisely. Its main job is to grab onto host cells, specifically by binding to a receptor called ACE2 on our cells, and then it orchestrates the fusion of the viral membrane with our cell membrane, letting the virus get inside.
The key that unlocks the door.
It's the key. And because it's the most exposed part of the virus and essential for that very first step of infection, it's the perfect target for generating neutralizing antibodies.
Neutralizing antibodies, the ones that actually stop the infection before it starts.
Exactly. The ones that physically block the virus from getting into cells. Studies on the original SARS virus had already shown that antibodies targeting the S protein were highly protective.
Makes total sense. But quick question, why not target other viral proteins like the nucleocapsid, the N protein? I know that one's really abundant too.
That's a great question, and it highlights the strategy. The N protein is very abundant, and it does trigger a strong immune response. But where is it located?
Inside the virus, with the RNA.
Exactly. It's inside, packaging the genome. So antibodies against N are great markers for telling if someone has been infected. But they can't actually stop the virus from entering a cell in the first place because they can't see the N protein until the virus is already inside or broken apart.
Ah, I see. So targeting S aims for prevention, blocking entry.
Precisely. They prioritized generating a protective, neutralizing antibody response, which, by the way, had a knock-on effect for diagnostics later.
How so?
Well, if you only test for anti-S antibodies, you can't tell if someone has them from the vaccine or from a natural infection because both generate anti-S. That's why anti-S-STAN tests were often used alongside, especially for tracking breakthrough infections in vaccinated people.
Got it. Okay, so spike protein it is. But just knowing it's the target wasn't enough, was it? For the best design, they needed to see it in, like, super high detail, atomic level detail. How'd they get that picture so fast?
They got it incredibly fast using a technique called cryo-electron microscopy, cryo-EM. It's been revolutionary for structural biology.
Cryo-EM, how does that work, basically?
So you take your purified protein, the spike protein in this case, and you flash freeze it incredibly quickly in a very thin layer of non-crystalline ice, like glass, vitreous ice. This traps millions of individual protein molecules in all sorts of random orientations.
Okay, frozen in place.
Then you stick it in a very powerful electron microscope and take literally thousands, sometimes hundreds of thousands, of 2D pictures of these frozen particles.
Just snapshots.
Right. And then comes the computational magic. Sophisticated software sorts through all these 2D images, figures out their different orientations, and reconstructs them into a high-resolution 3D map of the protein's structure. From that map, you can build an atomic model, see where every atom sits.
Wow. And what did those first cryo-EM structures of the SARS-CoV-2 spike protein show that was so important?
They confirmed it exists as a trimer, 3 copies of the protein bundled together, but the really crucial insight was seeing that a key part, the receptor binding domain, or RBD.
The part that actually grabs onto our cells.
Exactly that part. It can flip between 2 main positions.
A
Downstate, where it's kind of hidden or shielded, and an upstate, where it's exposed and ready to bind to that ACE2 receptor.
Down and up, okay.
Understanding that dynamic was absolutely critical. Why? Because an effective vaccine needed to train the immune system to make antibodies that could recognize and block the spike protein, specifically when that RBD was in the up, accessible, dangerous state.
Target the weapon when it's drawn, essentially.
That's a great way to put it.
Okay, this is fascinating. But I remember hearing there was another big problem. The spike protein itself is kind of unstable. It doesn't like to stay in that ready-to-infect shape.
You're right. That was a major hurdle. The spike protein is what's called a class I viral fusion protein. And these proteins are inherently metastable.
Metastable, meaning?
Meaning they exist in that high-energy pre-fusion shape, the shape before they trigger membrane fusion, but they're spring-loaded, essentially. They have a strong tendency to spontaneously snap into a much more stable, lower-energy post-fusion shape.
The shape after it's done its job.
Exactly. And here's the problem for vaccine design. Your immune system needs to see and make antibodies against the pre-fusion shape to block infection. But if you just produce the natural spike protein by itself in the lab, a lot of it will quickly collapse into that useless post-fusion shape.
So you end up showing the immune system the wrong target.
Precisely. It's like trying to train soldiers with pictures of exploded tanks instead of intact ones. Not very effective.
So how did they solve that? How did they lock it in the right shape? This sounds like real protein engineering.
It absolutely is. And this solution is a fantastic example of building on previous work. Scientists working on other viruses, particularly MERS-CoV and also RSV, respiratory syncytial virus, had already figured out that you could introduce specific targeted mutations to stabilize these fusion proteins in their pre-fusion state.
Ah, borrowing from other fights.
Exactly. The key insight developed by researchers, including Barney Graham's group at the NIH and Jason McClellan's group, was to use proline residues. Proline is an unusual amino acid. It creates a rigid kink in the protein backbone.
Like a structural brace.
Kind of like that, yeah. They realized that by strategically introducing 2 proline substitutions, replacing the original amino acids at positions 986 and 987 with prolines right in a flexible hinge region critical for the pre to post-fusion transition.
They could jam the mechanism.
They could effectively lock the protein in that desired pre-fusion conformation. This specific engineered version became known as the 2P stabilized spike protein P for proline, because there were 2 substitutions.
2P spike. Okay.
And this was a masterstroke. The 2P engineered spike is significantly more stable, it can be produced in much larger quantities, and crucially, it consistently displays that correct pre-fusion shape to the immune system.
And they confirm this?
Oh, yeah. Cryo-EM structures beautifully show that the 2p mutant was indeed stabilized in the pre-fusion state.
So let's connect this back to the listener. What does this clever bit of protein engineering actually mean for you and your immune system when you get the vaccine?
It means that the instructions in the vaccine tell your cells to make a version of the spike protein that looks almost exactly like the one on the surface of a live infectious virus before it attacks your cells.
You're right, mugshot.
The perfect mugshot. This trains your B cells, the antibody factories, to produce antibodies that are incredibly effective at recognizing the real virus and neutralizing it, grabbing onto it, and stopping it before it can cause infection. This rational, structure-based design, the 2p stabilization, is absolutely a cornerstone of the Pfizer-BioNTech vaccine success. It really elevates vaccine development to a true engineering discipline.
Amazing. Okay, so we've got our target antigen identified and perfected, the engineered 2p stabilized spike. Now, the core technology, how do you actually make the messenger RNA, the mRNA molecule, that carries the instructions for building that protein? And you mentioned this is totally cell-free.
That's right. It's a process called in vitro transcription, or IVT, and it doesn't involve living cells at all, but it actually starts with DNA.
Starts with DNA to make RNA.
Yep. You first design a DNA template, usually a circular piece of DNA called a plasmid. This plasmid is meticulously engineered using standard biotech tools, recombinant DNA technology.
What's in this DNA blueprint?
It has several essential parts arranged in a specific order. First, a strong start signal called a promoter, specifically one recognized by an enzyme called T7 RNA polymerase.
Okay, the go signal.
Then a 5' untranslated region, or a 5' UTR. It's a sequence that doesn't code for protein but helps stabilize the eventual mRNA and tells the ribosome where to bind efficiently.
Like instructions for the instruction reader.
Sort of, yeah. Then comes the most important part, the open reading frame, or ORF. This is the actual genetic code, the precise sequence of nucleotides that tells the cell exactly how to build that 2p stabilized spike protein.
The core message.
Yes, exactly. After the ORF, there's a 3' untranslated region, another non-coding bit that also boosts mRNA stability and translation efficiency. And finally, a sequence that codes for a long tail of adenine bases, the poly-A tail. This tail protects the mRNA from being chewed up by enzymes and also helps initiate translation.
So promoter, UTRs, the spike code, poly-A tail.
Yeah.
Got it. How do they make lots of this DNA template?
They grow this engineered plasmid in huge quantities inside standard laboratory E. coli bacteria. Then they purify the plasmid DNA. But there's one crucial step before making RNA. The plasmid is circular, and you need a linear template. So they use a specific enzyme, a restriction enzyme, to cut the circle open at a precise point right after the poly-A tail sequence.
Like unrolling a scroll.
Exactly. This linearization ensures that when the RNA is made, all the molecules stop at the same point and are the correct, uniform length.
Okay, linearized DNA template is ready. How do you actually make the mRNA from it? You mentioned IVT, in vitro transcription.
Right? In vitro just means in glass. So it happens in a reaction tube or vessel, not inside a cell. It's purely an enzymatic reaction.
Advantages to doing it that way.
Huge advantages for manufacturing. It's fast, it's highly scalable, you can make large amounts, and crucially, you avoid contamination with all the other stuff that's inside a living cell. You get a much cleaner product.
Makes sense. So what goes into this IVT reaction cocktail?
You need the purified linearized DNA template, obviously. You need a high concentration of that workhorse enzyme, T7 RNA polymerase. You need a buffered solution containing magnesium ions, which the enzyme requires to function. And you need a large supply of the 4 RNA building blocks, the nucleotide triphosphates, ATP, CTP, GTP, and airhead. Here's a key point. A modified version of uridine triphosphate.
Ah, the modified uridine again. We'll come back to that. So you mix all this together.
You mix it all together under the right conditions, typically a specific temperature. The T7 RNA polymerase finds the promoter on the DNA template, latches on, and then travels along the DNA strand, reading the sequence and synthesizing a complementary single strand of mRNA using those nucleotide building blocks. Zip, zip, zip.
Okay, now let's talk about that modification. The modified uridine. You called it a secret weapon. What is so special about it? Why the stealth modification?
Right, this is probably the most critical biochemical innovation behind the success of mRNA vaccines. For decades, a major roadblock for using mRNA as a medicine was our own immune system.
How so?
Our cells have built-in sensors, like toll-like receptors, TLRs, that are designed to detect foreign RNA, like from an invading virus. When they detect it, they trigger a strong inflammatory alarm and antiviral response.
Which is good if it's a virus.
Exactly. But if it's your therapeutic mRNA, this response is bad news. It causes cells to shut down protein production altogether, and it leads to rapid degradation of the very mRNA you just delivered. The medicine destroys itself and shuts down the factory.
A huge problem. So how did they solve it?
The breakthrough came from years of foundational work by doctors Catalin Carrico and Drew Weissman. They discovered something remarkable. If you replace the standard RNA building block, uridine, with a slightly modified version called pseudoridine,
Pseudoridine, okay.
the resulting mRNA becomes much less visible to those TLR sensors. It kind of flies under the radar of the innate immune system.
Makes it stealthy.
Precisely. And the Pfizer-BioNTech vaccine actually uses an even further optimized version called N1-methylpseudoridine, or M1 for short.
Well, M1A, got it. So how is it incorporated?
During that IVT reaction we just talked about, instead of adding regular uridine triphosphate, UTP, they completely substitute it with M1 triphosphate.
Yeah.
And luckily, the T7 polymerase enzyme is perfectly happy to incorporate this modified version into the growing mRNA chain.
Okay, so you get mRNA where every single U is replaced by an M1. What does this actually do?
This single, subtle chemical tweak delivers two massive synergistic benefits. First, immune evasion. The M1-attaining mRNA largely avoids triggering those TLRs. No big inflammatory alarm, no shutdown of protein synthesis, no rapid degradation. The mRNA gets to stick around and do its job.
Benefit number one, stealth. What's number two?
Benefit number two, enhanced translation. For complex reasons related to how the ribosome interacts with the mRNA and maybe the mRNA structure, this M1A modification makes the mRNA get translated into protein much more efficiently. Dramatically more protein is produced from each mRNA molecule.
Wow. So it hides better and works harder.
Exactly. More bang for your buck, biochemically speaking.
That is incredible. So how does that one modification ripple outwards? What's the impact beyond just that single molecule in a cell? Thinking bigger picture, like manufacturing, global health?
It's truly transformative. It elegantly solved the two big and historical hurdles for mRNA therapeutics, the inflammatory response and poor protein expression. And the ripple effect is profound because each mRNA molecule is now like a super productive little protein factory.
You don't need as much.
Exactly. You need a much, much smaller dose of mRNA to get the same or even better immune response. And that has huge implications for manufacturing.
Yeah.
A single production batch can now yield vastly more doses.
Which means faster rollout, potentially lower costs.
Right. It directly impacts the speed and scale of vaccination campaigns. It shows this amazing link between a fundamental biochemical discovery and the practical logistics of tackling a global pandemic.
Amazing. Okay, so the mRNA is synthesized with its stealthy M1 modification. Are there any final touches needed before it's ready to be packaged?
Yes, a couple of crucial finishing steps to get the pure mRNA drug substance. First, a special molecular cap has to be added to the 5' end, the beginning of the mRNA molecule.
A cap, like on a bottle.
Sort of. It's a chemically modified guanine nucleotide called 7-methylguanylate. This 5-foot cap does two vital things. It protects the mRNA end from being degraded by enzymes in the cell, and it acts as the landing signal for the ribosome, telling it where to bind and start reading the message.
Essential for protection and translation.
Absolutely. And nowadays in large-scale manufacturing, this capping is often done during the IVT reaction itself by including special cap analog molecules in the mix. Makes it more efficient.
Okay, capped. What else?
Rigorous purification. The raw mixture coming out of the IVT reaction isn't just the mRNA you want. It contains leftovers. The DNA template, the polymerase enzyme, unused nucleotide building blocks, and critically, some unwanted double-stranded RNA, dsRNA, byproducts can form.
Why is double-stranded RNA bad?
Because dsRNA is a major trigger for those same innate immune sensors we were trying to avoid with the M1 modification. So even tiny amounts of dsRNA contamination could undermine the whole stealth approach and cause unwanted side effects.
Ah, so you have to get rid of it meticulously.
Absolutely. Purification is critical for safety and efficacy. Usually involves multiple steps. First, an enzyme called DNase is used to chew up any remaining DNA template. Then sophisticated chromatography techniques are employed.
Chromatography, like separating colors.
Similar principle, but much more advanced. Techniques like ion pair, reverse phase chromatography are particularly good at separating out that problematic dsRNA from the single-stranded mRNA product. Often, this is combined with something called tangential flow filtration, or TFF. It's a way to simultaneously concentrate the purified mRNA and exchange the buffer it's dissolved in, getting it into its final formulation buffer. The end result is a highly pure, precisely engineered mRNA molecule ready for the next big step. packaging.
Okay, we have our perfect engineered stealth mRNA with its vital instructions. But you said earlier you can't just inject this naked mRNA. It's too fragile, and it can't get into cells easily. It needs a delivery vehicle. Enter the lipid nanoparticle, the LNP.
Exactly. Injecting naked mRNA is completely useless for two main reasons, as you said.
Yeah.
One, fragility. Your body fluids are packed with enzymes called RNases that would shred unprotected mRNA in seconds.
Gone before it even gets close to a cell.
Pretty much. Two, the cell membrane barrier. Both mRNA, with its phosphate backbone, and our cell membranes have a strong negative electrical charge. Like repelling magnets, they push each other away. Plus, mRNA is a big molecule. It just can't passively slip through that oily lipid bilayer to get into the cytoplasm where the protein-making machinery is.
So the LNP has to solve both problems.
It has to perform a dual role, yes. It needs to be a protective shield like armor, guarding the mRNA cargo from those destructive RNases in the bloodstream. And it needs to act like a Trojan horse, tricking the cell into taking it up, and then somehow releasing the mRNA inside the cytoplasm.
It strikes me this LNP isn't just like a simple bubble of fat. It sounds like another piece of sophisticated engineering, almost like nano architecture.
That's a perfect way to describe it. It's absolutely not just a simple fat droplet. It's a precisely engineered, smart drug delivery system. We're talking about a tiny sphere, maybe 80 to 100 nanometers across, that's incredibly small, thousands of times smaller than the width of a human hair. And it's constructed from a very specific recipe of 4 different kinds of lipid molecules mixed in precise molar ratios. This specific formulation isn't random. It's the result of literally decades of research and optimization in the field of nanomedicine.
Decades just on the delivery vehicle.
Yes, figuring out how to safely and effectively deliver nucleic acids like RNA into cells has been a huge challenge. This 4-component LNP design is a culmination of that work. It's cleverly designed to be stable and relatively inert while circulating in your bloodstream, but then to become active and perform its function once it gets inside a cell, specifically inside a compartment called the endosome.
Okay, break down the 4 components for us. What does each lipid do?
All right, let's meet the team. Component number 1 is the ionizable cationic lipid. In the Pfizer vaccine, this is a molecule called ALC0315. Think of this as the engine of the LNP.
The engine. How so?
It has a very special property. Its electrical charge changes depending on the acidity, the pH. At a slightly acidic pH, like during the manufacturing process, it's positively charged. This allows it to strongly bind to the negatively charged mRNA backbone, pulling it into the core of the forming nanoparticle.
Okay, binds the cargo.
But then, crucially, at the neutral pH of your blood, around 7.4, this lipid becomes electrically neutral. This is really important for minimizing toxicity while the LNP circulates.
So it's less likely to interact badly with things in the blood.
Exactly. But then, and here's the clever part, if the LNP gets taken inside a cell into an endosome, which naturally becomes acidic, this lipid becomes positively charged again.
Aha.
And that positive charge is key for the final step, escaping the endosome. We'll come back to that. So that's the ionizable lipid, the engine. It makes up about half the lipids in the particle.
Got it. Engine. Component 2.
Component 2 is a phospholipid, specifically DSPC in this vaccine. This acts as the scaffolding. Phospholipids are natural components of our own cell membranes. They help form the stable, organized lipid bilayer structure of the LNP shell, giving it integrity. Makes up maybe 10% of the lipids.
Okay.
Component 3 is cholesterol, another molecule naturally found in our cell membranes. Here it acts as a stabilizer or stiffener.
Cholesterol as a stabilizer.
Yeah, it's a relatively rigid, bulky molecule. It wedges itself into the lipid layers, filling gaps and increasing the packing density. This makes the LNP mechanically more rigid, enhances its overall stability during storage and in the bloodstream, and it also plays a role in helping the LNP fuse with membranes later on. It's a significant chunk, maybe around 40%.
Stabilizer. Makes sense. And the last one, number 4.
The 4th component is the pegylated lipid, in this case, ALC0159. This is the stealth cloak.
Stealth cloak. Like Harry Potter.
Sort of. It consists of a standard lipid molecule that anchors into the LNP surface, but it's attached to a long, flexible, water-loving polymer chain called polyethylene glycol, or PEG.
PEG? I've heard of that.
Yeah, it's used in many medicines. These PEG chains stick out from the LNP surface, forming a dense, hydrophilic, water-loving layer. This layer physically blocks proteins in your blood called opsonins from sticking to the surface of the LNP.
Why is blocking those proteins important?
Because those opsonins are like little eat-me signals for your immune system's cleanup crew, macrophages. If the LNP gets coated in them, it gets cleared from the bloodstream very quickly before it can reach its target cells. The PEG layer provides steric hindrance. It physically gets in the way.
So the stealth cloak hides it from the cleanup crew.
Exactly. This prevents the LNPs from clumping together and dramatically increases their circulation half-life, the time they can survive in the bloodstream to find their target cells. It's a small percentage of the total lipid, maybe 1.52%, but absolutely crucial for efficacy.
Ionizable engine, phospholipid scaffold, cholesterol stabilizer, and PEG stealth cloak. That's quite a team. So how do you get these 4 different lipids plus the mRNA to assemble perfectly into these tiny loaded nanoparticles? It can't be just mixing them in a beaker, right?
Definitely not just stirring them in a beaker. It requires a very precise, controlled, and highly reproducible manufacturing process, typically using microfluidics.
Microfluidics, tiny pipes.
Essentially, yes, extremely small channels etched into a chip. The process usually involves rapidly mixing 2 separate liquid streams. One stream contains all 4 lipids dissolved in an organic solvent like ethanol. The other stream contains the purified mRNA dissolved in an acidic aqueous buffer, maybe around pH 4.
Okay, lipids and alcohol, mRNA and acid water.
These 2 streams are pumped at very precisely controlled flow rates into a special microfluidic mixing chip. These chips have complex internal channel designs, like a staggered herringbone pattern, that force the 2 fluids to mix incredibly rapidly and chaotically within milliseconds.
Super fast, turbulent mixing in tiny channels. What does that rapid mixing do?
It triggers the self-assembly. As the ethanol containing the lipids rapidly mixes with the acidic water containing the mRNA, 2 things happen almost instantly. First, at that low pH, around 4, the ionizable lipid becomes strongly positively charged, remember?
Right, the engine turns on.
And it immediately forms strong electrostatic interactions with the negatively charged mRNA backbone, grabbing onto it. Second, the lipids are soluble in ethanol but not soluble in water. So as the ethanol gets diluted by the water, the lipids essentially crash out of solution they precipitate.
So they grab the mRNA and solidify around it.
Driven by these forces, the charge interaction and the insolubility, the lipids spontaneously self-assemble into nanoparticles, trapping the mRNA inside with very high efficiency, often over 90%.
Wow, it just happens automatically if you mix it right.
Under precisely controlled conditions, yes. The resulting mixture then goes through another purification step, usually that tangential flow filtration, TFF, again. This removes the ethanol and, very importantly, exchanges the acidic buffer for a physiological buffer around pH 7.4.
Why switch the buffer?
Because neutralizing the pH back to physiological levels causes the surface ionizable lipids to become neutral again. Remember, neutral in the bloodstream is good for minimizing toxicity. This step yields the final LNP formulation, ready for sterile filtration and filling into vials. And the use of microfluidics is key here for ensuring the LNPs are consistently uniform in size and have a high, consistent amount of mRNA loaded inside, crucial for quality and reliable dosing.
Okay, the LMPs are made, they're injected, they circulate thanks to their stealth cloak, then they get taken up by cells. You mentioned they end up inside a little bubble called an endosome, but the mRNA is still trapped in there, right? It needs to get out into the main part of the cell, the cytoplasm.
Yeah.
How does the LNP pull off that final escape?
Right, this is the final critical function of the LNP, facilitating endosomal escape. After the cell engulfs the LNP via endocytosis, this happens mainly in muscle cells near the injection site, but also importantly in professional immune cells like dendritic cells. The LNP and its mRNA cargo are indeed trapped inside this membrane-bound vesicle, the endosome.
So close, yet so far.
Exactly. But the cell itself helps out, unintentionally. As the endosome matures inside the cell actively pumps protons, hydrogen ions, into its interior. This is a normal cellular process.
What does bumping protons in do?
It makes the inside of the endosome progressively more acidic. The pH drops from neutral down to maybe pH 5 or 6.
Aha, the pH trigger again.
Precisely. This acidification is the specific environmental cue the LNP, particularly its ionizable lipid, is engineered to respond to.
So what happens when the endosome gets acidic?
As the pH inside the endosome drops below the turning point, the pKa, of that ionizable lipid ALC0315,
It becomes positively charged again.
bingo, it regains its positive charge. Now you have these positively charged lipids embedded within the LNP structure right next to the negatively charged lipids that make up the endosomal membrane itself.
Opposites attract or repel. What happens?
It's thought that these now cationic LNP lipids interact destructively with the anionic lipids in the endosomal membrane. The exact mechanism is still debated, but it likely involves disrupting the membrane structure, maybe forming pores, maybe causing the whole LNP to kind of fuse with or destabilize the endosomal membrane, leading to its rupture.
Creates an exit hatch.
Essentially, yes. The integrity of the endosome is compromised, and the LNP breaks free, releasing its precious mRNA cargo out into the cell's cytoplasm,
Where the ribosomes are waiting.
where the ribosomes are waiting to start making that spike protein.
It really is amazing. The LNP isn't just passive packaging. It's this dynamic, smart machine responding specifically to its environment to complete its mission.
That's exactly it. That pH-responsive behavior is the key. It ensures the LNP performs its final cargo release function only after it has successfully reached its target destination inside the cell. It's incredibly elegant bioengineering.
Okay, so the LNP has done its job. Engineered mRNA is successfully delivered into the cytoplasm. The vaccine's core mission is now underway. How does this mRNA actually hijack or maybe co-opt our own cells to build that immune defense?
Right, once it's free in the cytoplasm, the vaccine's mRNA molecule is immediately grabbed by the cell's own protein-making machinery, the ribosomes.
The cell's 3D printers.
Good analogy. The ribosome clamps onto that 5-foot cap we talked about. Then it starts moving along the mRNA strand, reading the genetic code, the open reading frame, 3 letters, nucleotides at a time. And as it reads, it recruits the corresponding amino acids and stitches them together in the correct order to build the protein.
And in this case, it's building the 2p stabilized SARS-CoV-2 spike protein.
Exactly. Your own cell could be a muscle cell where you got the shot, or an immune cell that picked up the LNP is temporarily turned into a mini factory churning out copies of this foreign viral protein. And thanks to that M1M modification and the optimized UTRs, this process is super efficient.
Lots of spike protein gets made.
Lots of it. And here's the crucial safety point. This is all happening based on mRNA instructions. The cell never sees the actual live SARS-CoV-2 virus. There's zero risk of causing COVID-19 infection from the vaccine itself. It's just making one specific engineered piece of the virus.
Okay, the cell is making spike proteins. Now, how does the rest of the immune system actually see these proteins and realize, hey, something foreign is being made here, sound the alarm?
That happens through a process called antigen presentation. The cell takes fragments of the proteins it's making, or proteins it engulfs, and displays them on its surface using special molecules called major histocompatibility complex, or MHC molecules.
MHC molecules, like little display stands on the cell surface.
Exactly. And the vaccine cleverly triggers presentation through 2 main pathways, mimicking what happens in a real viral infection. First, there's the MHC class I pathway.
MHC class I.
Okay. Some of those newly synthesized spike proteins inside the cell get chopped up into small pieces, peptides, by a cellular recycling machine called the proteasome. These spike peptides are then loaded onto MHC class I molecules, which are found on the surface of almost all your nucleated cells, basically everything except red blood cells.
So most cells put up this flag saying, look what I'm making.
Pretty much. It's like an internal status report. I am currently producing this protein. In real viral infection, this MHC class I presentation is the signal that tells killer T cells, this cell is infected, destroy it.
Okay, that's pathway 1. What's the other?
The second is the MHC class II pathway. This is mainly handled by specialized immune cells called professional antigen-presenting cells, or APCs. Dendritic cells are key players here.
Dendritic cells, the immune system's scouts.
Exactly. These APCs can either get directly transfected by the LNPs and make spike protein themselves, or they can gobble up debris from other vaccinated cells that might have died, or even engulf whole spike proteins that might be released. They process these proteins differently, breaking them down in internal compartments and loading the resulting peptides onto MHC class II molecules.
MHC class II, different display stand.
Different stand, different message. MHC class II presentation basically signals to other immune cells, I have found something foreign outside the cell or in my internal compartments. We need to organize a broader defense.
So class I says, I'm making this. Class II says, I found this.
That's a great way to summarize it. And having both pathways activated by the vaccine is really important. It intelligently mimics the immunological signals of a real infection, ensuring you activate the full range of your adaptive immune response, both arms.
Okay, the alarm signals these spike peptides on MHC molecules are being displayed. Who answers the call? Which parts of our immune army get activated? And what are their roles?
The presentation happens, and then these APCs often travel to nearby lymph nodes, the immune system's command centers. There, they activate the 2 main branches of the adaptive immune system, T cells and B cells.
T cells and B cells, the key players.
Right. First, you have helper T cells, also called CD4 plus T cells. Think of them as the generals or coordinators. When a helper T cell's receptor recognizes a spike peptide presented on an APC's MHC class II molecule,
It gets activated.
it gets activated, starts multiplying rapidly, proliferating, and releases chemical signals called cytokines. These activated helper T cells are crucial because they provide essential help instructions and stimulation to both the B cells and the other main type of T cell.
Okay, who are the other T cells?
Those are the killer T cells, or CD8 plus T cells. These are the foot soldiers of the cellular immune response. They get activated when their receptors recognize spike peptides presented on MHC class I molecules, so I'm making this, signals on regular cells, or APCs.
The ones that target infected cells.
Exactly. Once activated, killer T cells become cytotoxic effectors. They are programmed to patrol your body and seek out and destroy any of your own cells that are displaying that specific spike peptide on MHC class I. This is a critical defense for clearing out cells that might get infected if you encounter the real virus later.
Okay, so helper T cells coordinate, killer T cells destroy infected cells. What about the B cells? That's antibodies, right?
That's the antibody arm, yes. The humoral response.
Mm-hmm.
Some B cells have receptors on their surface that can directly bind to the intact spike protein itself, the whole protein, not just peptides.
Mm-hmm.
When a B cell binds the spike protein and receives that crucial help signal from an activated helper T cell,
It gets the green light.
it gets a green light to fully activate.
Right.
It then undergoes rapid proliferation and differentiation, turning into specialized cells called plasma cells.
Plasma cell.
These plasma cells are essentially dedicated antibody factories. They start churning out huge quantities of antibodies, specifically designed to bond tightly to the SARS-CoV-2 spike protein. And critically, many of these are neutralizing antibodies.
The ones that block infection.
And the ones that physically latch onto the virus's spike protein and prevent it from binding to and entering your cells.
Wow. So you get T cell help, killer cells, and antibody factories all firing up. That sounds like a strong immediate defense. But what about protection down the road? How does the vaccine set up long-term security?
That's the crucial role of immunological memory. While those initial high levels of antibodies and activated T cells will naturally decrease over weeks and months after vaccination,
The immune system doesn't just forget.
it absolutely does not forget. A key outcome of this whole activation process is the generation of a small but vital population of long-lived memory B cells and memory T cells.
Memory cells.
These cells are like veterans. They carry the memory of the spike protein. They can persist in your body for a long time, sometimes years, potentially even decades.
Waiting quietly.
Waiting quietly, but ready. Now, what happens if, down the line, you're actually exposed to the real SARS-CoV-2 virus?
The memory cells recognize it.
Instantly. These memory cells recognize the spike protein on the invading virus and launch a secondary immune response. And this secondary response is much, much faster, much stronger, and generally more effective than the initial primary response generated by the vaccine.
Like having troops already trained and mobilized.
Exactly. Memory B cells rapidly differentiate back into plasma cells and pump out massive amounts of high-affinity neutralizing antibodies. Memory T cells quickly reactivate, proliferate, and start hunting down any infected cells. This rapid recall response is what prevents you from getting sick or significantly reduces the severity of the illness if you do get infected. That's the core of vaccine-induced immunity.
That makes sense. And you mentioned earlier something about the LNP itself acting as a kind of mild alarm signal, an adjuvant.
Yeah, that's a really interesting and somewhat nuanced point. While the M1M modification brilliantly dampens down that unwanted excessive inflammatory response from the mRNA itself, the LNP delivery vehicle, and perhaps the mRNA structure to some extent, still provide a sort of gentle danger signal to the immune system.
Not enough to cause problems, but enough to say, pay attention.
Exactly. It acts like a mild built-in adjuvant. An adjuvant is something added to vaccines to help stimulate a stronger immune response. This mild intrinsic adjuvancy of the mRNA LNP platform helps to efficiently activate those antigen-presenting cells, like dendritic cells, and ensures you get a robust adaptive T cell and B cell response, all without needing to add a separate, potentially more reactive adjuvant compound.
So it's a finely tuned balance.
It's a very clever, nuanced balance, enough stimulation for a strong response, but muted enough to maintain a good safety profile.
We've covered so much ground. We've reached the end of this deep dive. And looking back, it's just crystal clear that the Pfizer-BioNTech vaccine wasn't some overnight fluke or isolated event. It really was this incredible scientific synergy, wasn't it?
It absolutely was. A landmark achievement, no doubt. It truly represents the convergence of multiple powerful technological streams that had been maturing, often independently, there for decades. You had the rapid genomics for sequencing. You had structure-based rational protein engineering to perfect the antigen. You had the incredibly advanced mRNA biochemistry with the M1A modification. And you had the sophisticated smart nanomedicine of the LNP delivery system.
Each a field in itself.
Each a major field built on countless hours of dedicated, often unglamorous basic research. And then the pandemic provided this intense, urgent focus that pulled all these threads together.
Looking forward, what do you think is the biggest single takeaway from this success story? For me, it keeps coming back to the platform itself, the mRNA-LNP combo, and its incredible modularity.
You've hit the nail on the head. That modularity is the real game changer for the future. The fundamental architecture, the process of making modified mRNA via IVT, the four-component LNP formulation, the manufacturing process, it's largely antigen agnostic.
Antigen agnostic, meaning you can just swap out the message.
Pretty much. The genetic sequence encoded in the mRNA, the open reading frame, can be easily changed or swapped out. If you want to target a different protein from a different virus or even a cancer antigen, you just change the DNA template used for the IVT reaction.
Like changing the software in the same hardware?
That's a great analogy. The core hardware, the mRNA production and LNP delivery system, largely stays the same. We saw this in action with the rapid development of updated COVID boosters targeting new variants. They didn't have to reinvent the whole vaccine technology, just update the spike sequence.
So it's like a plug-and-play vaccine platform.
Exactly. And that positions mRNA technology as this incredibly powerful platform for future pandemic preparedness. Imagine a new pathogen emerges. Scientists sequence its genome. They identify the key target antigen. Within weeks, potentially, they could design the mRNA, synthesize it, formulate it in LNPs, and have a clinical-grade vaccine candidate ready for testing. Not years, weeks.
That is just revolutionary potential. But surely there are still challenges. It can't all be smooth sailing from here.
Oh, absolutely. Challenges remain. Scaling up manufacturing to meet global demand is still a huge undertaking. You need secure supply chains for all those specialized raw materials, the enzymes, the modified nucleotides, those specific lipids for the LNPs. Developing robust, efficient, large-scale purification processes is complex.
And the cold chain, right, storage.
Yes, the ultra-cold storage requirements for these early mRNA vaccines pose significant logistical hurdles, especially in resource-limited parts of the world. Research is ongoing to develop more thermostable formulations, but that remains a challenge.
So work still to be done.
Definitely. But despite those challenges, the success of BNT162b2, the Pfizer-BioNTech vaccine, has irrevocably changed the landscape of vaccinology and medicine. It provided definitive, large-scale validation for the entire mRNA-LNP platform. It's not just about COVID-19 anymore. This technology is now being actively explored for vaccines against many other infectious diseases, flu, HIV, RSV, Zika.
And beyond infections too, right?
Yes. Huge potential in oncology cancer vaccines designed to train the immune system to attack tumors. And even for treating rare genetic diseases by delivering mRNA that codes for a missing or faulty protein. We are truly witnessing the opening chapter of a new era in biomedical science, an era defined by incredible speed, precision, and frankly, unprecedented potential.
What an incredible journey. We've gone all the way from that first patient sample through the atomic level engineering of the spike protein, the ingenious stealth mRNA, the smart nanoparticle delivery system, and finally seen how it masterfully orchestrates this multi-layered immune defense right inside your own cells. For you listening, hopefully that gives you a real shortcut to understanding what is truly a revolutionary technology.
It's really layers upon layers of innovation.
It really is. And just think about this as you go about your day. How many different, seemingly unrelated scientific fields had to develop and mature, often quietly in the background over decades, just waiting for this moment, this global need, to suddenly converge and create something as transformative as this vaccine? It's mind-boggling.
It truly speaks to the power of long-term investment in basic science.
Absolutely. We really hope this deep dive gave you some of those aha moments, connecting the dots. Keep asking those big questions.